Oh. Hello, could somebody confirm you can hear me?
I can hear you.
Thank you. Sorry for being late, but the Zoom must have downloaded an update. And when I went to Zoom, I didn't know where I was and I couldn't get to my lecture page on Zoom. So I had to scroll around and look up my account number and all that, and that's why I'm a little late. Are there any questions for today? If there's no questions, let me state that I've got a lot of grading done. I think all of the labs are graded except for lab 9, and then the people who turned in late assignments. The late assignments I have not gotten graded yet, but otherwise lab 0 to lab 8 are graded. The last lecture worksheet, and then I guess every worksheet before that has been graded. And I finally graded all of the quizzes. I have not gone in and manually corrected the score on quiz 5 yet. And I haven't dropped the quiz yet. So I haven't figured out which quiz to drop and then bring up quiz 5 so that it's worth the total number of points. Any questions about any of that? Any questions about anything? All right, we'll take a look at the grading. There's still at least one major project I haven't started on yet, and that's your infectious disease project. And I hope to get grading on that at least by this weekend, if not sooner. All right, if there's no questions, let's begin. Let me share my screen. So today is November 29th, and we are supposed to be finishing chapter 13. I don't know if we will do that, but I'll certainly try. There is no lecture for today. I will be logging in for 15 minutes from, 6:30 to 6:45 to find out if there's any questions. You do not need to show up unless you have questions. And then after 6:45, if there's no further questions, I'm going to log off. Anything else about the lab I should talk about? The unknown projects is due Saturday, December 3rd. That is coming up. Is that this is this Saturday? Most of you have already identified your unknown bacteria species, but there's at least a few of you who are still working on it. So you want to get going on that. I think there's one student I need to send a what do you call it, the results to today, but as far as I know, there's just one student that I'm going to be sending results to today. Remember, your unknown notes pages in the lab will be closing before Saturday. They close on Friday, and so you want to update those pages on Friday. A lot of you have not put in the results and your conclusions at the end. Make sure you do that. The unknown notes page is worth 5 points. It's not a lot, but it is worth some points. And then let me last state that there will be a final on Wednesday, December 7th. You must finish that by 8 p.m. Let me see if I can look into that. Make sure that's true, but I think most of the quizzes I set for, you could start them on Tuesday at 8 p.m. and it must be finished by 8 p.m. on Wednesday. Well, there's no way I'm gonna know how to log in here. So let me try another browser and see if I can get it. That might work. What is this? This one will log in nearly automatically. There we go. Can you guys see my pages? Can you see my computer? Nobody's saying anything. All right,
Yeah,
I'm showing
yeah, we can see it.
critical because I'm afraid you might be able to see it. There will be 2 parts to the final, and let me make sure that it ends when I said it did. Where did that go? Oh, there it is. Yep, now I lost it again. Just a minute. Let me get that. Yeah, you can start it Tuesday, December 6th at 8 p.m. It must be finished by Wednesday, December 7th at 8 p.m. Just wanted to make sure that was right. There are 2 parts. Part A, just a few questions. They're fill in the blank or short essay. And then most of it will be part B, which will be the multiple choice questions. There's about 50 multiple choice questions. And let me see, where is it? It does require Respondus Lockdown Browser for the multiple choice questions. The part A does not, and once you start on it, as long as you finish by 8 p.m., it's fine. Make sure you are finished by 8 p.m. because Canvas will lock you out if you do not finish by 8 p.m. Any questions about the final? It is going to be comprehensive, but there will be heavy coverage of chapters 7, 13, 14, and lab module 9. These are material that you have not been quizzed on. There will be questions from all the other chapters and all the other labs, but generally there will not be more than about 1 or 2 questions from each chapter. The exception is chapter 1 because it's an introductory chapter. We run into the topics in chapter 1 throughout the textbook. And so if there's a question in chapter 1, you saw it in chapter 1 and some later chapter. And so there might be more questions from chapter 1. But all of the other chapters and labs shouldn't have more than about 2 questions on the final. Except for, of course, like I said, the last chapters, which you have not been quizzed on. And that would be chapter 8, chapter 13. A little less on chapter 7 because we're only going to cover that for part of next Thursday. Any questions about the final? All right. If not, I think I'm going to close that because I won't need it for the lab. Let's go ahead and get started on the lecture. So we had talked about growing bacteriophages in the lab, and when you grow them on a petri dish, they form plaques wherever there's a virus, and that's because it's a lytic virus we're looking at here. and it lyses the bacteria, creating the plaques. Any questions about any of that? All right. So when we're talking about viral multiplication, the viruses contain only a few genes. Their genome only has a few genes, such as the capsid proteins. If they have specific enzymes they need, it might be in them with the virus. Although the viruses do tend to use enzymes that are in the cell already, but sometimes they have specific enzymes that they use and they'll be carried along the viral genome. So how the virus multiplies is they infect their host cell and hijack it, and then use the host cell's metabolic machinery, such as the host ribosomes, host tRNA, host amino acids, host nucleic acids, the host ATP, and some of the host enzymes to replicate the virus. The basic mechanism for viral multiplication is similar in all viruses. We're going to be talking about viral multiplication happening first in viruses that infect bacteria cells, and these cells or viruses are called bacteriophages. There are viruses that infect bacteria. Bacteriophages follow two multiplication mechanisms, and we call these life cycles, how the virus replicates. The first bacteria viral life cycle is the lytic life cycle, where the host cell that becomes infected is lysed and killed, and then that releases the newly replicated viruses. The other way that bacteriophages can multiply is following the lysogenic life cycle, or the, we call it the lysogenic life cycle. And the virus does not kill the host cell. The host cell remains alive, and the two live pretty much in harmony, the virus and its infected host cell. At least as while the lysogenic life cycle continues. So before we talk about viral multiplication, let me ask, what do we need for a bacteriophage to replicate? What needs to be replicated? What does the virus need? Anyone have any guesses? All right, well, we need to replicate the viral genome. If the viral genome is double-stranded DNA, that has to replicate into copies of the viral double-stranded DNA. And then the DNA has to be transcribed into messenger RNA. The messenger RNA has to be translated mainly into the capsid proteins, but there could be some other proteins. And then the capsid proteins has to get together with the viral genome to make the new progeny virions, the new viruses. And this is something that happens for all viruses. I'm showing it only for double-stranded DNA viruses, but it would be similar for all viruses, regardless of what its genome is. It's just, obviously, if it's an RNA virus, and it's a positive strand, single strand RNA virus, you don't need to transcribe it because that genome can be used as messenger RNA. And then you have to translate that to make the capsid proteins. And once again, the capsid protein has to get together with the viral genome, which does need to replicate, even though it's messenger RNA, it does have some form of replicating. And we'll talk a little bit more about that in a little bit. Any questions about any of that? All right, the lytic life cycle follows 5 stages for the life cycle. First, there's attachment, where the virus literally attaches to the host cell. Then there's penetration, where the virus injects its genome into the host cell. Then there's biosynthesis, where the virus genome is transcribed and translated, and the virus makes DNA, or if it's a DNA virus, makes copies of its genome and then makes its capsid proteins. And then there's maturation, where the bacteriophage particles assembled into new virions. And then there's release, where the virus releases itself from the cell, and how that happens is it digests the cell, and we'll talk a little bit about that. That kills this host cell, by the way. So in the lytic life cycle, the host cell dies. Let's watch a little movie about this. If I can get this to run. I think this now runs. This should work. Why isn't that working? For some reason it isn't working. Yep, I don't have the address there. See if that works. Oh, shoot. I have a feeling this one won't work on this computer.
First, the phage particles stick to the surface of the bacterial cell.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay, any questions about any of that? We'll watch the rest when I talk about the lysogenic life cycle. All right, let me see if I can go back. Here it is. I think I've talked about this. Let's move to the next slide. So initially in the lytic life cycle of a bacteriophage, the virus has to attach to the host cell. There are specific attachments, like a virus like T4, for example, bacteriophage T4 will only attach to E. coli, and it has to do with the ligands on the molecule of virus, which are also called adhesions, molecules that attach to their host cell. And for this type of a virus, the attachment site is actually on the lower part of the tail fibers, meaning it's this part of the tail fiber right here and here. And this specifically interacts with receptors, what the virus binds to, specific molecules on the host cell. And that is we call a receptor. Most of the adhesions or the attachment sites of the virus, as well as the receptors of the host cell, are glycoproteins. Any question about any of that? Doesn't have to be a glycoprotein. I'm just saying most of them are glycoproteins. That's the first part. I guess this is all I have. So then what happens is the virus actually contracts. So like that, it contracts like that. That puts pressure on the contents inside the head, which is the nucleic acid. And when it contracts like that, that pushes the nucleic acid in the head through the hollow tail and into the cell. Now, I guess I haven't talked about this, but the base plate will get rid of the cell wall. And what it does is it uses the enzyme lysozyme, which digests the peptidoglycan in the cell wall. And then without the cell wall, it's much easier to inject something into the cell, and the tail is designed for doing this, injecting the nucleic acid of the virus into the host cell. Any questions about any of that? Like I said, it's the contraction that forces the nucleic acid in the head into the tail, much as a syringe. When you push on it, it's the contraction of the syringe pushes the contents of the syringe into the patient. Anything else I want to talk about that? No. All right, let's move on. Next, that's called penetration. So there's attachment, then penetration, injecting the nucleic acid into the virus. The first thing a lytic bacteriophage does is, once its genome is inside this host cell, the first gene transcribed is a gene that actually degrades the host chromosome. And I think I mentioned this before. Why this happens is the virus wants to call the shots in the host cell that it infects. And if it degrades the genome of the host cell, then the viral genome is calling all the shots of what's going on in the cell. And then the viral genome is replicated, making new copies of the viral genome, and the viral genome is transcribed and translated to make the capsid proteins. And on this virus, we have at least 3 capsid proteins, maybe even more, because there would be at least one for the head, and then probably 2 actually for the sheath of the virus, and then a third for the tail fibers. And then there's probably another protein in the base plate. So there's at least 4. Anyways, those are all made in biosynthesis. I'm supposed to have another slide here. Where the heck is that? Go back here. And then there's maturation, where the phage particles assemble. Yeah, I'm supposed to have another slide here. I don't know what happened to it. The viral particles actually self-assemble, and they're designed to just come together. There is an enzyme which stuffs the viral nucleic acid into the head, and then the rest of the virus self-assembles. And then the last gene of the virus that's turned on is lysozyme, which codes for an enzyme which digests the cell wall of the bacteria cell. And with that gone, water will move into the cell, and the cell will eventually burst. As enough water moves in, the cytoplasm will eventually burst, and that will release the new virions, the newly replicated viruses. Any question about any of that? All right, two terms we need to know with the lytic life cycle is burst time. That's the time from when the cell is initially infected with the virus to the time where the cell burst. And depending on the temperature, this averages 20 to 40 minutes. And also depending, I guess, on what virus species we're talking about. The burst size is the number of newly released viruses. It's usually between 50 and 200. Once again, it depends on the virus species. Any question about any of that? All right. The other viral multiplication life cycle in bacteriophages is the lysogenic life cycle. In the lysogenic life cycle, the host cell is not killed. Upon attachment of a lysogenic phage to the host cell, it will go through attachment and penetration. And then one of two things can happen. A lysogenic virus can enter the lytic life cycle, in which case it'll go through the lytic life cycle that we just discussed. Where do I have that? Right there. Okay. Meaning go through attachment, penetration, biosynthesis, maturation, and release. And it'll happen the same way. Release will be by digesting the cell and killing the cell. So a lysogenic virus can enter the lytic life cycle. Any questions about that? The other thing that can happen is once the lysogenic virus enters the cell, it can enter the lysogenic life cycle. In the lysogenic life cycle, the virus does not kill the cell, and actually the virus doesn't replicate on its own, and it does replicate with the cell, but other than that, so the lysogenic virus stays alive, if that's the correct term, stays in the host cell, does not kill the host cell. And then that begins the lysogenic life cycle. The phage remains inactive, and how that happens is, well, I'll talk about that later. There's a lysogenic protein that keeps the lytic life cycle inactive. We'll talk a little bit about that. And the bacterial host cell is turned a lysogenic cell. It does remain alive for as long as the virus is in the lysogenic life cycle. Any questions about any of that? All right, so let's look at that again. Once the lysogenic virus infects a host cell, one of two things can happen. And the virus has to decide what it does at the start. It can enter the lytic life cycle, in which case it just goes through the lytic life cycle, replicates itself, bursts the host cell, releasing the lytic, the virus, I guess the lysogenic virus here, which can then go out and infect another cell. The other thing the lysogenic virus can do, and it decides this immediately once it infects a cell, it can enter the lysogenic life cycle, in which case the lysogenic genome of the virus will integrate into the host chromosome. Any questions about that? When we have the viral genome, which will be DNA in this case, because it's entering the DNA chromosome, it will be a physical part of the chromosome. And this, remember, a circular chromosome. So just inserts itself into that circle. And then we have the viral genome right there with the host genome right there. And the viral genome is called the prophage at this point. While the virus is in the lysogenic life cycle, it will remain a prophage, integrated into the host cell chromosome. The only time the viral genome can replicate is when the host cell replicates its genome. So when the host cell decides to replicate, it will replicate the host chromosome, but it'll also replicate the prophage. And then we'll have two daughter cells, each with the prophage. Each will be in the lysogenic life cycle. This can continue for many divisions. Every once in a while, for reasons which are not well understood, and they've been studying this for over 100 years, and they still don't have a good understanding of what happens, but the lysogenic virus, which is entering the lysogenic life cycle, will excise itself from the host chromosome, no longer a prophage. And then we have the viral genome out here and the host cell chromosome here. And what happens is when this happens, the lysogenic life cycle discontinues and the lytic life cycle then will begin. So a virus which is in the lysogenic life cycle can switch to becoming in the lytic life cycle. And that switch, like I said, is not well understood. They do know that if you damage the host cell's genome, like expose it to UV radiation or some other type of radiation, you can switch this cell from a lysogenic life cycle to a lytic life cycle. And that's probably just because the virus knows this cell is likely to die, and so it's going to replicate with a lytic life cycle before the cell dies. But for other reasons why it's switching, like I said, it's not well understood. Let me say that they do understand why the lysogenic life cycle will continue and the lytic life cycle is shut down. And that is, there is a repressor protein in the lysogenic life cycle. Where do I have that here? Repressor protein, and it keeps dormant the lytic life cycle genes. So as long as this repressor protein is acting, the cell will remain in the lysogenic life cycle. Any questions about any of that? So one term to remember, prophage, when the lysogenic life cycle, lysogenic virus is in the host cell's chromosome. And I guess I've already talked about that. All right, any questions about the lysogenic life cycle? All right, if there's no questions, let's talk about some of the consequences of lysogeny. First, a lysogenic cell, meaning a lysogenic bacteria cell, is immune to infection by viruses of the same species. Another consequence of lysogeny is phage conversion, where the host cell can exhibit new properties from the genes in the bacteriophage. So here we have the bacterial chromosome, and it obtains new DNA from the lysogenic virus, the lysogenic viral genome, which becomes a part of the host chromosome. These genes can be expressed, and that can change the phenotype of this cell from that cell. Any questions about that? One example of phage conversion where the cell changes its phenotype because of the genes of the lysogenic virus happens with Corynebacterium diphtherium. Corynebacterium diphtherium does not produce the diphtherium toxin unless the bacteria is infected with a lysogenic virus, meaning the toxin of Corynebacterium diphtherium is encoded by the virus. So yes, it will depend how sick you will become from a viral infection on whether or not that bacteria, did I say viral infection? I mean bacterial infection. It will depend on whether that bacteria is infected with a lysogenic virus or not. Corynebacterium diphtherium will not produce the diphtherium toxin unless that bacteria has the lysogenic virus within it. Any questions about any of that? All right, if there's no questions about that, that's enough about talking about bacteriophages. Let's talk a little bit about animal viruses, how they replicate and I guess infect cells, which would be similar. The multiplication of animal viruses follows the basic pattern for bacteriophage multiplication, but there are several differences. For example, there's a difference in the attachment. Animal viruses only attach to the cell membrane because animal cells don't have a cell wall. Penetration can differ, and we'll talk a little bit about that. It happens by either endocytosis or by fusion. There can be uncoating of the virus that's similar to a bacteriophage. No, I guess it isn't similar because the bacteriophage injects its copy into the host cell, and the animal virus will uncoat to release the viral genome. Biosynthesis is similar to bacteriophages. Maturation is similar. Release is different. The release can happen by a rupture of the cell, and that we call the lytic life cycle of an animal virus. So animal viruses can follow the lytic life cycle, the same one as a bacteriophage following the lytic life cycle. We do not have a lysogenic life cycle for animal cells, but we have something a little similar. It's obviously a lot different too, but and that's the budding life cycle, where the budding virus lives in harmony with its infected cell. The budding virus does not usually kill the host cell, and then the cell will bud out the new viruses, why it's called the budding life cycle. Any question about any of that? We talked a little bit about enveloped viruses. An enveloped virus follows the budding life cycle, and you'll see why once we talk about the budding life cycle. All right, let's talk just a little bit about multiplication of animal viruses. I've discussed already the multiplication of animal viruses follows about the basic pattern of multiplication of bacteriophages, with one major difference. Bacteriophages can only replicate in the cytoplasm because in a bacteria, that's all there is. In an animal virus, they can multiply in the cytoplasm, but they may also replicate in the nucleoplasm. So that's the main difference. And I'll go ahead and state, since I already mentioned this, we'll talk a little bit more about it. But if an animal virus replicates in the cytoplasm, A rule of thumb is it's an RNA virus. A rule of thumb is a rule that applies to almost all viruses. There might be a few exceptions, although for this one, I don't know of any exception where a DNA animal virus replicates in the cytoplasm. I don't know of any. Okay. And then for the rule of thumb, it's if it's a virus replicating in the nucleus, the rule of thumb is it's a DNA virus. There is one exception, but we'll talk about that exception. But DNA viruses replicate in the nucleus. Why? That's where all the animal cells components for replicating DNA are kept. And so the virus replicates in the nucleus because it's a DNA virus and it wants to replicate its DNA. And so it wants to use the host cell machinery to replicate its DNA genome. So it needs to go in the nucleus to replicate. Any question about that? So remember those rule of thumbs. All right. The multiplication of animal viruses, the attachment happens to the cell membrane. Penetration of the virus doesn't happen by the virus injecting its nucleic acid into the cell. Penetration happens by entry of the virus, either by endocytosis or by fusion. If you have an old version of the textbook, it will erroneously say entry is by endocytosis or by fusion. Excuse me, not by endocytosis. The old version of the textbook will say pinocytosis instead of endocytosis, and that's not correct. Okay, it happens by endocytosis. Endocytosis is where something attaches to the host membrane of the cell, and then the host membrane invaginates, and then this will pinch off, meaning this will, I don't know if you can see my fingers here, pinch off where the cell membrane will connect and go right across, and then this will pinch off and connect right across, and that will make the vesicle for this virus being brought into the cell. Any question about endocytosis? Here we're looking at it in a little more detail. The virus attaches to its receptors. This invaginates further. This will then pinch off where this membrane will connect to that membrane. This membrane will connect to that membrane. And then the vesicle will be degraded, as well as the virus will be degraded to release the viral nucleic acid into the cell. That is by endocytosis. It can also happen by fusion. Let me see if I can show you this. Let me see if I got that on this computer. Oh, I was supposed to show you that, the rest of that, sorry. We'll go back to that. That I can shut down. See if I can get paint up here. I don't have it. Okay, let me shut this down just for a minute. Come on. Not getting it. Oh, here it is. Never mind. Stop the share. Here we go. And what am I looking for? I'm looking for a picture so I can open up Paint. If you're wondering why I'm shutting it down, it's because there was a student who complained once when I opened up a picture on my computer. It was a perfectly fine picture, And she said it was pornography, and they looked at it and there was no pornography on my computer. And I just decided I'm not going to deal with any of that again. And so you're not going to see what I'm showing there, which is definitely not pornography because I have no pornography on my computer. But all right, we got paint here. So let me show this again. Oh, having a hard time reestablishing the share. There we go, share screen. All right, so you can see my screen, is that correct? Let's bring this down here.
Yes.
Great. I want a brush. All right, so this is a little virus. That's not what I wanted. Why isn't that showing up? There's a brush. Oh, there we go. A little virus. And then this is going to be a big cell. The two come together. I should have made that bigger, sorry. The two come together, and how do I do that? We'll just erase this one. I'm going to have a hard time drawing this, so we're just going to assume this is a double membrane. Maybe I can draw it with the virus. There we go. And then this will be a double membrane here too. Not drawn very good, but you get the picture. Well, they confuse right here. That's probably not the right place to do it, but it goes like that. And then that's actually connected right there, and that's connected there. And except there's a problem. Let me see what the problem is. I can do this drawing a little better. Yeah, that's actually not bad. Connect this one right here to there. And now this one to there. Sort of. Can you sort of see that? And that's the fusion, where the fusion of the viral envelope, an envelope around the virus, fuses to the cell membrane. And then that will fuse together where the viral envelope will become a continuation of the cell membrane. And so the cell membrane actually goes around. Draw that a little better. Now we got it. Goes around and then around the virus and then back around the cell again. And that will open up, and then the contents of inside this virus can then move from inside the virus to inside the cell. That's fusion. It happens with a viral envelope and the cell membrane. Any question about any of that? You guys should have a question, and that is, why is that happening? It's because the viral envelope is very similar to the cell membrane. They're mostly it's mostly a lipid bilayer, the envelope. Any questions about any of that? All right, no further questions. Let's move on. I did mention about the multiplication of animal viruses. It can happen in the cytoplasm, in which case a rule of thumb is it is an RNA virus, or it can happen in the nucleus, and that will be a rule of thumb, a DNA virus. There is one exception, and that is retroviruses. They are an RNA virus that replicate in the nucleus. And we'll talk a little bit about that exception, and you'll understand why that's an exception once I talk about retroviruses. All right, didn't move. All right, let's talk a little bit about the multiplication and release of the viruses. The viruses can be released from an animal cell by the lytic life cycle, where the virus actually lyses the host cell, and then that releases the newly replicated viruses. I think the human cold follows the lytic life cycle? I should actually look that up because I've never, never looked it up. Another virus that I know that follows the lytic life cycle is the polio virus. It follows the lytic life cycle. I think most naked animal viruses follow the lytic life cycle. I think most do. If the virus is an envelope animal virus, it follows the budding life cycle. Any question about that? The budding life cycle is another life cycle we only find in animal cells that I'm aware of. It's possible that some protists have viruses that infect protist cells that follow the budding life cycle, because there are some protists which are similar to animal cells. But I don't think there's any plant viruses or even fungal viruses that follow the budding life cycle. So for us, we're just going to say it's an animal virus that's enveloped. What happens is the virus replicates, and the replication of the virus, if it's following the budding life cycle, is there aren't many viruses replicated at one time. In fact, in the entire lifespan of the cell that's infected, there aren't that many viruses that are replicated. The virus will make a copy of itself, and it has to make its viral genome and its viral capsid proteins. And then the virus self-assembles. And then the virus comes up to a membrane. This can be the cell membrane as shown here. It can be the nuclear membrane, or it can be the membrane of the endoplasmic reticulum. It depends on the virus. Okay, what membrane it comes up to. Most viruses take it from the cell membrane, but like I said, it can be from the nuclear membrane, or it could be a few viruses even take it from the endoplasmic reticulum. The capsid protein with the viral genome comes up to the membrane, and it'll move to a portion of the membrane that has viral proteins and glycoproteins in it. Meaning the virus genome will make a protein or a glycoprotein, and then that protein will be put into the membrane. The virus will come up to a part of the membrane that has the viral proteins in it, and then it'll push on that membrane, and then push out tremendously on that membrane, making a bud. And then it'll push further, ripping off that bud, which will self-seal, and then the membrane itself will also self-seal. Okay? Any question about any of that? The part of the membrane that's ripped off will now form the envelope to the enveloped virus. And that's how a budding life cycle virus obtains its envelope. It literally rips off a piece of the membrane of its host cell. And like I said, it could be the cell membrane, it could be the nuclear membrane, or it could be the membrane of the endoplasmic reticulum. Here we see a budding virus forming buds on a cell. I assume that's the cell there. So that's an actual electron microscopic image, and there's more. Any questions about budding viruses? Any questions about why the envelope of a budding virus is similar to the cell membrane? It's because it is part of the cell membrane. The exception is it can have some viral proteins and glycoproteins in it. All right, viruses have different genomes. There's double-stranded viral DNA genome, single-stranded DNA genome, a double-stranded RNA genome, and a single-stranded RNA genome. Therefore, each virus type needs to have a slightly different mechanism for multiplication and biosynthesis. It's very easy to multiply double-stranded DNA because you just use one strand as a template to make a copy of the other strand. And actually, that's the same for double-stranded RNA. You use one RNA strand to replicate and make the other strand. So that actually is similar. We'll talk a little bit more about single-strand DNA replication and especially single-strand RNA replication. We'll talk a little bit more about those. This chart, I know you can't see it well. Let me see if I can blow this up. Is showing you the family of DNA viruses that infect humans and cause disease. There are 7 families of viruses that cause disease in humans, at least that we know of. And this is telling you the virus family name. You don't need to memorize this, the type of DNA it is, whether it's an envelope virus or a naked virus, some things about the virus, and then the diseases that they cause, like smallpox, for example, is a member of the Poxviridae family. I think you know about herpes viridae family, the herpes virus. There would be herpes simplex type 1 and type 2, for example. We talked a little bit about the herpes viruses. I will talk a little bit about adenoviruses, I think. And that might be it. Maybe a little bit about smallpox. I don't remember if we talk about vaccine or not. All right, let's end the show here. You don't need to memorize the names of the DNA viruses. I'm just showing you that there are 8 families that cause disease in humans. And these are DNA animal viruses. Let's talk a little bit more about the DNA animal viruses. They contain DNA for their viral genome. The DNA animal viruses usually have the DNA copied in the nucleus using viral enzymes, meaning the DNA polymerase is actually encoded by the viral genome. The capsid and the proteins are synthesized, though, in the cytoplasm. So that means the DNA, the viral DNA, has to be transcribed, and then that transcript moved to the cytoplasm. And then it uses the host cell's machinery for translating the proteins. Multiplication of the DNA virus. One of 2 things can happen once viral DNA enters the host cell. The DNA can form a provirus where the DNA of the virus literally integrates into the host chromosome. How that virus will replicate is when the host cell replicates its DNA, it will replicate the viral DNA. A difference between the animal DNA virus that integrates in the host chromosome and the bacteriophage DNA that integrates into the host cell chromosome is if the animal virus DNA, if it integrates and becomes a provirus, not a prophage, but a provirus, it will remain permanently a part of that host cell's DNA for as long as that host cell is alive. The viral DNA will never be excised from that host cell. Any question about any of that? But it can make copies of itself to replicate the virus. And like I said, the virus can also be replicated when the host cell replicates its own DNA and then separates into two cells. Each cell, each daughter cell will get a provirus. The other thing that can happen is the viral DNA can be expressed, producing new virions. The double-stranded DNA viruses will replicate their double-stranded DNA, and then the DNA will be transcribed. The messenger RNA will be translated into capsid proteins. The capsid protein will get together with the replicated DNA of the virus to make the new progeny virions. Any question about any of that? So this is showing you the multiplication of a DNA virus. It will infect a host cell, attaching the receptor sites of the host cell, and then that virus will come into the cell either by endocytosis or by fusion. The viral DNA will be released. The viral DNA will move to the nucleus because this is a DNA virus. It'll move to the DNA, the nucleus, and then the viral DNA can be transcribed to make viral messenger RNA, which will move into the cytoplasm to be translated into viral protein. The viral genome can be replicated so that we now have several copies of the viral genome. The viral genome can then get together with the capsid proteins, making the newly replicated virions, which can then be released from the cell, either by lysis of the cell if the virus is following the lytic life cycle, or the virus can bud out of the cell if the virus is a budding life cycle virus. Any question about any of that? All right, that's the multiplication of DNA viruses. Now let's talk about RNA viruses. You'll note that there are more families of RNA viruses that cause disease in humans. There's about 15 families of RNA viruses that cause disease in humans. We'll talk a little bit about Orthomyxoviridae, very little about it. Retroviridae, we'll talk about them. We'll talk about Coronaviridae. And then we'll talk a little bit about herpes viridae. I will mention that Picornaviridae is the virus family that includes smallpox, as well as polio. Do I have that right? I might not have that right, that's the one having smallpox. I'll have to look that up. I think smallpox is an envelope virus, in which case it's not Picornaviridae because it's a naked virus. Anyways, Picornaviridae includes the rhinoviruses. These are viruses that are the main cause of the human cold. The second most common cause of the human colds are the family Coronaviridae. These are coronaviruses. Yes, the one that encodes COVID-19 is also Coronaviridae. And then the third cause of the human colds is actually a DNA virus. I forgot to mention that. Come up here. Adenoviridae. There are members of Adenoviridae family of viruses that also cause the human colds. So there are 3 viral families that cause the human cold. And countless species, don't know how many species, but 3 viral families. And did I mention that it's polio, which is what I was trying to think of? It comes from the Picornaviridae family, not smallpox. Smallpox comes from Poxviridae, which can't read it here. I don't remember which family Poxviridae is in. I'll have to look that up. All right, here we're looking at some animal viruses. You'll note that they are very heterogeneous, and there's obviously more families that cause human disease than DNA viruses. Multiplication of RNA viruses is similar to the multiplication of DNA. There are some differences because we're starting with an RNA genome instead of a DNA genome. Most RNA viruses replicate in the cytoplasm. There is one exception, and that's the retroviruses. They replicate in the nucleus. And there are some differences in the RNA virus. For example, if it's a positive strand, single strand RNA animal virus, it'll replicate a little differently than the negative strand RNA animal virus. So DNA will transcribe it into messenger RNA, translate it into protein. How we copy the DNA is we just use, it's not shown here, we use one strand as the other to copy it, and then we get both strands of DNA copied. For RNA viruses, if it is a positive strand, single strand RNA virus, you can use that genome of the virus as messenger RNA to translate the viral protein. To copy the viral genome, what you do is you make a negative strand RNA virus, or a strand of RNA, and that will be the complement to the positive strand. And then you use the negative strand as a template to make more of the viral genome. And that's how you replicate positive strand, single-stranded RNA viruses. If, on the other hand, you have a negative strand RNA virus, you take the negative strand genome and you make a positive strand of the RNA, and then you use that as a template, the positive strand, to make more of the viral genome, the negative strand RNA. Any questions about any of that? Now, for the negative strand viral genome, you can't read that in the ribosome. So what you do is you make the positive strand RNA corollary to the negative strand, and then you use that positive strand as the viral messenger RNA to translate protein. Any question about any of that? Now, it's not shown up here for double-stranded RNA virus, but for double-stranded RNA virus, you just do the same thing, and that is you use one strand of the RNA as a template to make the other strand, and then you can replicate double-stranded RNA. For double-stranded RNA, you only use the positive strand of the double-stranded RNA to make messenger RNA, and then you use that to translate and make your proteins. Any question about replication of RNA viruses. All right, this is just showing you again, if you got a negative strand viral genome, you replicate that viral genome by using the negative strand as a template to make positive strand, and then use the positive strand as a template to make more of the genome, the negative strand genome. You use the positive strand to make the capsid proteins, you translate that, and then the capsid proteins get together with the newly replicated viral genome to make the progeny virions. Oops, go back. Or a positive RNA virus genome, you use that to make the negative strand RNA, and then you use this as a template to make more of the viral genome, the positive strand, single strand RNA. Remember, only the positive strand RNA is transcribed to make the capsid proteins, which will then get together with the replicated viral genome to make the progeny virions. All right, this is showing you a viral infection for a, let's see if I can blow this up. For a positive sense RNA virus, it will attach to the cell. The virus will be brought into the cell. The RNA genome, which is positive strand, will be copied into negative strand, and then the negative strand will be used to make more of the viral genome. The positive strand will be used to translate proteins, which will make the capsid proteins of the virus. It will get together in maturation to make the newly replicated virions, which will either then be released from the cell following the lytic life cycle or the budding life cycle. Any questions about positive single strand RNA virus multiplication? All right, negative strand, single strand RNA virus replication happens similar. The virus will attach to the cell, be brought inside the cell. The difference is the negative viral genome will be used as the template to make the positive strand of the virus. The positive strand of the virus will then be used to make the negative strand, which is the viral genome. Only the positive strand will be transcribed into viral proteins to make the capsid proteins, which will then get together with the newly replicated viral genome to form the new virions, which will be then released from the cell by lysis if the cell or the virus is following the lytic life cycle. If it follows the budding life cycle, the virus will be released by budding. All right, any questions about any of that? 2 minutes. Well, we get started with retroviruses. I won't finish it, unfortunately. The multiplication of retroviruses is a little different. They are an RNA virus, Retroviridae. However, they do not follow the normal rule of thumb. This RNA virus replicates in the nucleus. And if you understand about retroviruses, you'll know why they replicate in the nucleus. Retroviruses include HIV-1, HIV-2, and which cause AIDS, as well as feline leukemia virus, which causes feline leukemia, which is a disease similar to AIDS in cats. It's not AIDS, but the leukemia causes the cat to weaken its immune system, and then the cat's immune system finally gets shot when the cat gets very sick from the leukemia, and then the cat usually gets an infection. So the immune system is shot, and the cat usually will die either of the infection or of the leukemia. Retroviruses are different because they contain a gene for the enzyme reverse transcriptase. Reverse transcriptase is an RNA-dependent DNA polymerase. What reverse transcriptase does is it makes DNA from an RNA template. In fact, retroviruses come from the abbreviation of reverse transcriptase, RE from retrovirus, RE from reverse, and then TR from transcriptase. All right, I think I'll stop here. If there's no further questions, we'll continue with this. Yeah, I've got quite a bit more to go in this lesson. We'll continue here on Thursday. Any questions? All right, if there's no questions, those of you who have questions about the lab, I'll be there from 6:30 to 6:45.